welcome to everyone, and thank you very much to Miguel for having accepted to be here today. So Miguel Rocha is an associate professor at the University of Minho in Portugal, where he is director of the Master in Bioinformatics. He is also a senior researcher at the Center of Biological Engineering, where he leads the Bioinformatics and Systems Biology research team, an interdisciplinary group with around 20 researchers. And he has been the PI of several research projects and coordinated several open source software projects, and he has also a number of collaborators. Over the last years, he has focused his research on the exploration of two distinct but complementary topics. The first is systems biology, where different modeling paradigms have been used to optimize metabolic systems using evolutionary computation, with applications in metabolic engineering, but also recently in cancer research. And the second is machine and deep learning, with the development of algorithms and tools to handle different types of data, omics data, literature, compounds, and protein sequences. And that is being applied to distinct biomedical applications. So, Miguel, the floor is yours.
Thank you, Marie-France. Thank you for the invitation, and also Susanna for inviting me to present part of our work here. And thank you, of course, also for the nice introduction. So I'll start sharing. So let me know if anything doesn't go well. So I will today present some of our work mainly in constraint-based modeling of human metabolism, and start a bit where we came from also in metabolic engineering, but trying to see how we can combine these ideas in constraint-based modeling with AI. And as Marie said in the introduction, we are a group in the Center of Biological Engineering at Minho, where we have a team of around 20 people, and this started as a collaboration between computer scientists and biotechnology. The part of the work, or most of the work that I'll present here, have been made with this group, and that is a very interdisciplinary group with very different collaborators, and with the funding of FCT, EU, and some private companies. And also, of course, we are in a small city in the west part of Europe, so we need to collaborate with many people around the world to try to make meaningful work. So this is a bit a map of some of our current collaborations. As was said in the introduction, so in our group we try to work in what we call synergetic skills of two different areas, AI and systems biology. And we are a research group that started and still does lots of work in constraint-based models, and that's mainly what I'll discuss here. But we try to encompass this research into let's say a vision of research that is a bit summarized in these three rectangles. So this stack of technologies and scientific methods, and our idea is that we can have good models of our systems, and I will here mainly focus on models of cells, so larger systems, so that we can be able to predict in this case mainly predict phenotypes of cells. And our main goal is in the group has always been to try to use these two first layers to then do optimization. And in this scenario, optimization will be optimization of cells, both for biotechnology purposes like metabolic engineering, and also in biomedical applications. So this is our view. Then we try, of course, to solve some problems using this view. Today I will focus a bit on how to optimize cells so we can reach phenotypes of interest. But we also work in the group, and then happy to discuss this with anyone that's interested. We also work a lot on novel pathways, novel transformations, pathway design, and also a bit more recently, but also with some work on how to develop novel compounds, and we have an architecture to try to develop compounds with desired biological properties and activities that also incorporates a lot of machine learning and evolutionary computation approaches. Okay, so but today I'll focus in this in red, so the cell optimization, and I will focus in two main applications in our group, which are metabolic engineering, because it was where we started, and many of the ideas come from our work here in strain optimization. But also I will talk a bit about some recent work more on the human side in cancer metabolism. And so just discussing a bit the three layers here, and I will assume that most of the audience has some background on constraint-based models, but I will still try to explain a bit some of the principles that we use. And so everything starts with the model reconstruction. And so this is mainly a bioinformatics, let's say, pipeline with then that is also complemented with lots of experimental validation in an iterative cycle. And most of the ideas here come from taking a genome, annotating genomes, and from that creating portfolios of reactions that may occur in the metabolic systems of different organisms. And from there, with the help of lots of literature and experimental validation, try to fine-tune these reaction portfolios with different information regarding, for instance, the compartments, reversibility, thermodynamics, and energy requirements, biomass equations, and so on. So these are all steps that need to be done well for the model to work, and that are less automated than the annotation part. From there, we get a metabolic model that I will discuss. Just to say that we have a tool for anyone that is interested. We have a very nice tool for metabolic model reconstruction, which has as a main feature that it allows manual curation with a very nice user-friendly environment. And so it has been improved over the years. The last version 4, was this year also republished in NAR, and it is an actively developed tool that I invite everyone to try. And with that, we have in our group been working in many different models in different types of organisms. So you can see that the tool really helps on this model reconstruction part. Okay, so moving a bit to the second layer of how we can use these models to do phenotype simulation. And here I will try to be very brief in explaining what is this constraint-based models, because that probably everyone already knows a bit about that. But the idea is to define the cell and the boundaries of the cell, to create a matrix where you represent reactions and metabolites, and then using some assumptions for our models, mainly the steady state assumption, we can define constraints over this representation. The main constraint of constraint-based models is typically the steady state, which assumes that internal metabolites in the cell will remain with equal concentrations. And so this allows to create constraints over the fluxes of the reactions in the system, equaling the sum, of course, with the given signs of the reactions. The sum of these reactions that create and consume a given metabolite will need to be 0. Then we can add different types of constraints, and I think one of the major strengths of constraint-based models is that you can represent very different constraints, biological constraints in your system. For instance, you can have reversibility constraints, you can have bounds for the different reactions, you can have constraints that come from regulation, and so on. So there are a plethora of different approaches then to add new constraints over this system. But still, in most cases, we are normally left with underdetermined systems, so we don't have a clear way to solve these systems of equations to get the exact fluxes. And so we are left with normally two choices. One is to just use this to constrain parts of the space and then use a bit algebra to study those null spaces, so the spaces where all constraints are valid, or if we want to have specific flux distributions, normally we resort to using this as constraints and then applying optimization, and that of course needs optimization functions. And flux balance analysis is one of the main methods here, probably the most ancient one, where we typically assume that we want to maximize an artificial flux which stands as a mathematical representation of biomass in our system. So what we are trying to mimic here is to maximize growth of a given cell, and this is quite nice mathematically because it's a linear objective function. Since we have linear constraints as well, this works well with linear programming to be easy enough to solve and to get a flux distribution. Still, this has some problems since the flux distribution is normally not unique, so there's still a space of optimal solutions, and it assumes that organisms were evolving over time to maximize biomass. So in a way, we are saying that organisms are tuned to maximize growth, which is not always the case, mainly when you are then trying to impose genetic conditions over a given organism, for instance, trying knockouts or over and under expression phenotypes. So a different number of alternatives have been developed, for instance, to simulate mutants. These Mom and Room methods are quite used, where the idea is instead of maximizing growth, we are trying to minimize the flux difference of the distributions from the wild type and the mutant that we are simulating. And this is also interesting because it allows not only to use wild type simulated, for instance, from FBA, but also to use experimental data that we have on fluxes, for instance, for a given strain as a reference for the wild type. And so this model then can be enriched with several layers of information. For instance, these gene-protein reaction rules are quite common in metabolic models. I would say that probably most of the, or all of the recent models have this layer. And here what we try to represent is how we get from a gene to a reaction. And this is typically put as Boolean rules. Some models have all these layers, the gene, peptide, protein, reaction. Some models only have a simplified version with genes and reactions. And we are able to study a bit what is the influence of, for instance, knocking out a gene in the set of reactions that we have in our model. And this allows that models can work with different types of also omics data in these layers, for instance, of transcriptomics and proteomics. Also, there have been some efforts in trying to put regulation into models. Now we'll try to discuss a bit this in the remaining of this talk. But the idea, or the most simple idea, is that we can add some equations in this level of the GPRs. We can also add Boolean equations for transcriptional regulation, and this can work to impose then constraints over the metabolic system. Okay, so moving a bit to the part of optimization. So with the constraint-based models and these methods for phenotype prediction, we have a way to predict what can happen if we start changing the genes and the reactions in our system. And so what we try to do, and this is a work that started many years ago in our group together with many others in the communities, to try to think, can we use these ideas to improve the way metabolic engineering is done? Can we create strains in a more rational way? And first of all, we need to optimize strains, because typically wild type strains, although they have in many cases potential to produce a given metabolite, they are not optimized to do that as we desire in industrial strains. And our idea was, can we predict computationally what will happen in certain modifications in our genome, so we can create optimized cell factories in a rational way. So the idea is that if we can predict computationally what will happen, we can design optimization methods that will be able to design new strains, and then we hope that they can work in the lab. So we started doing that mainly with this bi-level optimization approach, where we have the inner level, which is basically phenotype simulation. So we call this like the cellular objective. So the idea of the inner level is to simulate as good as we can what will happen to that strain, and then we have an outer layer which will impose modifications that will then be tried, let's say, in the inner level, and then we can define a bioengineering objective, for instance, to maximize the production of a given compound of interest in industry, and so we can try which of the sets of modifications will work best. Okay, and so we did lots of work in this part, trying to optimize different things like gene deletions over under expression, what we can insert or not in host organisms from different organisms. All of these things are a bit different. They can, in some cases, be combined, but the idea is always to try to see what will be the best modifications that will work in our in silico approaches. And then, of course, at some point we need to go to the lab and validate, so do the modifications that the in silico part is recommending, and try to see if they really work or not. Okay. And so we did this with different approaches, with different models. We use a lot of evolutionary algorithms to optimize this part, and then we eventually started also to incorporate different modeling approaches. One of the modeling paradigms that appeared in the last years was the idea of adding new constraints that came from the enzyme usage in our models. So these GECKO models are one of the approaches in this direction, and the idea here is that we can put more information into our constraints, into our models. In this case, considering the enzyme as part of the reaction, and working with enzyme pools as some of the variables in our model. So in here you can see what happens to a simple reaction A to B. In this case, adding the enzyme, and this implies that our matrix grows. So we end up with some columns that will represent the enzyme usage, and some lines that will represent the enzyme pools in our model, and we can impose constraints individually in each reaction or in each enzyme, or we can, in some cases that we don't have data to do that, we can have more global constraints that will consider the total of protein of enzymes and the limit that to a given value. So this is of course a different approach. In our group, we have been developing this MuPy tool, which is let's say a metabolic engineering tool, but that is flexible enough to encompass other applications, and that basically tries to run both the phenotype simulation and the optimization part for different types of models. You can see here some of the methodologies that we have in our models. It works with different technologies in terms of what we can use for the simulation part. So we try not to create new things when they are available in other packages. And we have our main scientific contributions in the optimization part, where we define different types of multi-objective approaches. And in here, the multi-objective makes sense because in many cases we want strains, for instance, that can overproduce a compound, but also that will be able to grow at least at a minimum level, or we want to minimize the number of modifications that we do, and so on. So there are a number of different objective functions in the optimization layer that we try to encompass. So this mu-pi, the idea is that we have different optimization algorithms. We have different methodologies to simulate the phenotype, so we can compare them both, and with this we can generate different strain designs using this plethora of methods, both in the phenotype simulation and in the optimization layer. Okay, so moving a bit to what probably my title would entice, this set of methods is also being applied in health or human health related themes for many years. I would say that the three main areas are related with cancer, with pathogens, where we try to study in most cases the host pathogen interactions, and of course, and this would be expected also with many metabolic related diseases or conditions like diabetes, obesity, inborn metabolic errors, aging, Alzheimer, and so on. So there are a number of different approaches in all these areas, which have been quite interesting, although, and I'll probably finish there, in this area is still much less developed than the metabolic engineering applications, since also the cells and the systems are much more complex and much more difficult to design. It's much more difficult to design methods that really can simulate these systems. But the main rationale here, and I think this is important, is that we can promote precision medicine in this case because we are in principle able to fine-tune the different metabolic models using omics data to different groups of patients and to different genetic backgrounds. Okay? And so this is I would say, the driver for this type of research. And this, of course, needs models, and for a while now, I think the first published models for human metabolism come from 2007, so they are turning 15 years this year. And so for the last 15 years, there's been a number of different models. I'll say that the most used now and the most recent is the human one model that came out 2 years ago, and that has been improved over these 2 years also with different versions, and that has now around 13,000 reactions and about 3,600 genes. And now the community has of course evolved, so there is version control, there is lots of collaboration in trying to improve this model, and so things are gradually improving in this part. One important thing is that these models are general purpose models for human cells, so they encompass all the reactions that may happen in all human cells. So they are very general, which can be an advantage or a disadvantage, as we will see. There was some initial work on the human models using these general purpose models in different areas of the ones that I mentioned, like the ones that are put here, including even a cancer cell metabolism that is generic. Okay, and then this showed a number of limitations in these approaches, because since it is a very general model, it was difficult to simulate it in specific tissues, cell types, and the objective of these cells, these different human cells, is not to grow. So there is also problems in what can we use as objective functions for our phenotype simulation methods. And of course, different cell types have different metabolic objectives, which makes more difficult to create methods that can work for different things. And then two different approaches arise that gradually are actually converging a bit, which is the first one is to simulate based on what we know, for instance, in terms of omics data, to try to have specific methods for the simulation of specific conditions or cell types. And the other one is a bit different, as to say that we should start by having models for each different type of cell, and then using those models as a basis for simulation. So they are similar, but they are a bit different in the way that they approach this. But they converge in one thing, is that mainly they use what we know from the different conditions, both from literature and from the data that we have available, mainly omics data. And this is actually used to develop the objective functions that will be used in these two cases, because both of them will imply at some point to run optimization. So we need to define what is the objective of that optimization. And normally the objectives are based on these things. Okay? So there are a number of methods that arise to do this tissue-specific reconstruction and the phenotype simulation, and they have different ways to look at the objective functions and the algorithms. Some of them focus more on the idea of keeping reactions that are essential to keep some metabolic tasks, so some things that we know specific cells do to try to keep them working. Others are more based on looking at, for instance, gene expression thresholds and working, saying that if a given reaction is encoded, needs a specific enzyme or set of enzymes, then we can look at the proteomics data or the transcriptomics data from the genes associated to those enzymes, and we can define thresholds to say if that reaction should be working or not in a given tissue or a given cell type. And then other methods rely more on the idea of core reactions that can come from literature or extracted from data analysis pipelines, where we say, okay, these reactions will be core, these reactions will be non-core, not so important to our models, and then work from there. All these three have in common that we have a template, so we have a structure in our model that comes from the generic model, and that structure is always used to keep the models obeying all the constraints from the constraint-based approaches. So all these things here are layers that we put over that constraint-based core that is the basis of the methodology. Okay, and you see here some taxonomy of some of the methods that I mentioned. So this is only one example, so I will not I will skip a bit this. And so what we get is that we reinforce the number of different applications that we could do with this, and these are much more specific in the types of problems that are being addressed, like obesity, aging, diabetes, and so on. And this each work with different methods and different types of cells. Okay, and this also allowed to reinforce the applications in cancer. And cancer has been important because we know that the basic idea of FBA kind of makes sense with cancer cells, because they are one type of cell that we know that it makes sense, at least in some cases, to think that it is trying to grow as fast as it can, or to divide as fast as it can. And so the cancer was always a major theme in in this constraint-based models, as we see from some of this early work. So this is not very recent, but it was early work on cancer and specific types of cancer, using these methods that allow to create specific tissue-specific models. Okay. So now I can show you a bit of some recent work in our group related to cancer. And in this work, it's very recently published in PLOS Computational Biology. We try to systematize a bit some of the work that has been done in this area. So there is a lot of discussion if these methods are able to really predict well fluxes, to predict phenotypes, if they are really useful or not, for instance, in drug discovery. And so we try to make a more systematic, large-scale study of this, and then in a second part to extend a bit what model representations we can use to get more layers into our system. So this first part was published. This second one is still not published, so it's with new data here. And what we did in the first part was to do some variations of both what are the ways that we can pre-process omics data, and this is a very important technical issue in the field, how we should use, for instance, RNA-seq data and what kinds of pipelines we should use to pre-process those. And in the part of the reconstruction, what are the best algorithms that we can use to have those tissue-specific models for different cell types and how we can use. And then, of course, we try to validate this in different ways and to see what we could get out of these things. To do this, we use two in-house tools, which I also invite you to try. There are two Python frameworks that allow to create tissue-specific models based on omics data, and that implement many of the existing algorithms in the literature. So these are mainly Tropo, which is the one that allows to do this kind of work. So as I said, we try different approaches for the pre-processing. I'll not go into details here. We of course needed to do this, all the different ways that we can go from transcriptomics data to the reactions and the fluxes. And so this needs also some different ways to calculate this based on the GPRs of the models. I can go if you want to have some questions on this. We did a method based on elementary modes to gap fill the models, so to make sure that all models were able of producing biomass, so we could run simulations over these models. And we then use this CCLE data sets that have different types of omics data, that have knockout screens, that have also metabolomics data. So we use these data sets to run our approach. And actually, what we did, so we try to use as validation the essentiality of genes, fluxomics, so data on fluxes that were measured, and of course phenotypes from literature. So these are the ways to validate this. So what we did was to take the RNA-seq that they have in CCLE for each cell line, which are around 1,000, I think, and then do some pre-processing, try different ways of integrating this data into the models and trying different combinations of these methods. And we started doing that with a subset of the whole CCLE database for all the models for breast cancer, for the MCF7 cell line. And so we use this because it was a case where we had fluxomics data, and our approach was to run the pipeline, try the different approaches of our pre-processing algorithms with this smaller data set, see what works best, and then taking those setups and running in a larger data set. Okay, and so we run this for this MCF7 data. We looked for fluxomics from this paper from Rupin's group at the time in Israel. Now he's moved to the US. And we took the fluxomics for 44 reactions and used this to see how our model did. And so we had mainly two different phenotypes, which are gene essentiality predictions and the fluxes. In gene essentiality, our approach worked very nicely, so we were able to get some MCCs that were in line with the published data from the human one model, and in our best cases a bit higher. So it fit well what we knew. In terms of predictive fluxes, we realized that in many cases our fluxes that were being predicted were only marginally better than random predictions. So the flux prediction was not really working very well. And we realized that the growth media is a very important aspect in this. So no one really knows what is a media where cell lines need to be to be simulated in silico, because no one really knows in many cases what is in the medium that the cells are using in vitro and in vivo. And so we then took the best approach, the best setups, and run for 700 CCLE cell lines. So overall, we reconstructed more than 6,000 models, and then we did some tests. For instance, we tried all the breast cancer data and see if we still could keep some of the features that we know from the gene expression data. For instance, in the molecular subtype of the different cancers, and we see that although the flux predictions are not that good, it still keeps some of the structural information in the data. But when we use these flux predictions to predict, in this case, in a machine learning application, which in this case is predicting primary disease, we realize that the fluxes don't work well when compared with using the model structures and the gene expression data initially. Okay? And so we tried a different approach. So this is still ongoing work that has not been published, although we already have some results. And so what we did in this work was to try to create an integrated modeling approach where we could have some information, for instance, related to the different levels of the omics data and the different biological levels. So we try to incorporate here regulation, signaling, and of course the GPRs and the enzyme levels. But everything, and this is of course a limitation of this modeling approach, everything is done with linear equations. So we have here some assumptions that can be oversimplifying the system. And so I will go. I think I'm not sure how much more time I have, if I have.
Actually, yes, indeed, we are reaching the end, but you can go on a little bit more. We reached already the end of the talk, but you can go a little bit over.
Okay, so I will just. So then if there is some question, I can go back here. So this is the details on the framework, and say, as you see here, we have some assumptions, for instance, that enzyme availability is correlated linearly with fluxes, or that it is correlated with gene expression. And the idea here is that we have pools of the different molecules in our system, and the overall system is a very enlarged matrix where we impose linear constraints, and then we try to incorporate data in our system. And so this is the overall formulation for this. And basically what we saw is that calculating fluxes based on this new approach really improved things. So we can see here much more clearly the different molecular subtypes in breast cancer, and we can see here that our machine learning prediction, although it's still a bit worse than the gene expression data, it really improves, and it is much closer to these predictions. So since I'm already without time, but I can just give you some ideas of what we are doing. So one of the things that we are trying to do is use the idea of evolutionary algorithms that we came from strain optimization to try to do drug target discovery. So the idea is that we can try to find, for instance, gene knockouts that work well in killing, let's say, cancer cells, but to keep the normal cells alive. Okay. We are also working with Leiden in LUMC in some modeling of the tumor microenvironment with the work of Sarah. Sorry, that we are trying to model cells of the immune system and cells of tumors. And so this is some details on this work. And with the work of Tanya's PhD, this is actually published. We have some tissue-specific models for human cancer stem cells, and so we try to study a bit what we can take out of these models. So I'll not enter the details here. So to finish, I will just mention that although I think the constraint-based models are promising approaches in many different health-related subjects, they are still very much in their infancy, and there's still much to do. And as we saw, probably the first thing to do is to try to understand why flux predictions in many cases are still so bad, and how we can try to improve those. And our work in trying to get new layers that allow to put all the omics data that we have available on our models, I think are the way to go. Of course, not only with this formulation, but with many others. So we have GECKOs, we have ETFL models, we have ME models, and so on. So all these new modeling approaches, I think, will give another view of this, and will improve the results in our community. So thank you all. I'm sorry for exceeding time.
Thank you very much, Miguel.